Thank you very much, Jessica. My name is George Belibasakis, and I'm professor of clinical oral infection biology here at the Department of Dental Medicine. The title of my talk is the microcosm of oral biofilms. The main discipline of my research since the beginning of my career has been oral microbiology, and in particular how different members of the oral microbiota can cause or can be involved in causing common oral diseases. The term oral microbiota refers to collectively to all microorganisms that reside in our oral cavity. They have co-evolved together with us during evolution in a symbiotic relationship, and they are recognized primarily as commensals by our immune system. We know nowadays that we have more than 700 oral bacterial species in our oral cavity, and approximately half of them are as yet non-culturable. But more recent methods have demonstrated that we can have actually up to 19,000 or even more different phylotypes. What is special about our oral microbiota is that it tends to form complex biofilm communities on the tooth surface. So biofilms are complex polymicrobial communities which are embedded in their own polymeric matrix. Within biofilms, the individual bacteria can become more virulent compared to their planktonic or free-floating form. And as a result, they are less sensitive to components of the immune system, such as phagocytes or antibodies, immunoglobulins, and less susceptible also to antimicrobials. Here is an example of an in situ biofilm from an earlier study. Here is the potentially the postulated surface of the tooth, and you can actually see densely colonized bacterial populations. As we actually move outwards, the diversity of the species increases. The spaces between the microorganisms is filled by their own extracellular polymeric matrix. And at the very outer surface, when the biofilm has maturated, chunks of the biofilm can actually be cleaved off and be swallowed. So this is the result of an abstinence of oral hygiene for 4 or 5 days or so. When it comes to biofilms forming on the tooth surface, we have two main types: the supragingival and the subgingival biofilms. Supragingival biofilms are biofilms that are formed above the gingival margin, and subgingival biofilms are formed within or below the gingival margin. And they have different ecological conditions or different characteristics also. You can't really see clearly the subgingival biofilm, but it has grown in this space between the tooth and the gingiva, and as a result, the gingiva respond by producing inflammation, which you can actually see the initiation of the inflammation here. The most common oral diseases are actually of biofilm etiology. We have periodontal diseases, gingivitis and periodontitis. Gingivitis is the first initial inflammation of the tooth surrounding gingival tissue. If gingivitis or if the inflammation progresses deeper into the periodontal tissue, that would actually cause their destruction and lead to periodontitis. We have also dental caries. Dental caries is the result of the demineralization of the hard tissues of the tooth as a result of acids produced by bacteria in the biofilm that are able to ferment carbohydrates. If dental caries become deeper, they would eventually reach the pulp space of the tooth and may cause pulpitis and a necrotic pulp. And if this infection is spread further deeper outside of the apex of the tooth, then that will result to apical periodontitis. We also have oral fungal infections, stomatitis. These are more frequently associated with denture wearers or with immunocompromised patients, or they are an outcome of increased antibiotic resistances. And last but not least, we have a more modern disease or more series of modern diseases, peri-implant diseases, mucositis and peri-implant mucositis and peri-implantitis. These are the result of the application of dental implants, which I wish I would have more time to talk about it in this lecture, but that's perhaps a completely separate topic. I will use periodontal disease as a model for my talk here in relation to biofilms, because I've extensively worked on that and I think it's a great model. In periodontal health, our gingiva are pink, have a stippled appearance that looks like the surface of an orange, and they are not bleeding. And the gingival margin has a knife-edged appearance. If biofilm accumulates on the surface of the teeth and remains for some time, that will eventually cause an inflammatory reaction by the gingival tissue, which you can actually see here by light bleeding. This is an initial protective, so to speak, response. But if it is prolonged over long periods of time, and if the biofilm cause is not removed, then that may eventually progress deeper into the periodontal tissues to cause periodontitis, which is the destruction of the alveolar bone and the supportive alveolar bone and connective tissue, and eventually to tooth loss if the disease remains untreated. Periodontitis, therefore, is the main cause of tooth loss in adults, and it accounts for 10 or 50% of the world population, really depending on the severity that we are looking at. The prevalence increases by age. Clinical characteristic is the periodontal pocket and bleeding. The periodontal pocket is really a great environment for the formation of an anaerobic biofilm. And during surgery, when we raise the flap, we can actually see the loss of bone. The bone should have been actually close to the cervical region of the tooth, but it has been destroyed as a result of the inflammatory process. That comes also with microbiological changes of the associated biofilms. In health, we have gram-positive bacteria, anaerobic and non-motile. But when a biofilm becomes subgingival and is associated with periodontitis, the dominating bacterial species are gram-negative, anaerobic, and motile. It's quite simple and straightforward, actually, to sample biofilm from the tooth surface. So we're quite lucky when we deal with dental research, because it's fairly easy to collect clinical samples. Dental biofilm, supragingival or subgingival, can actually be collected fairly easily, either with a curette, which is an instrument that we use in treatment, in periodontal treatment. The biofilm can be scrubbed off supragingivally or subgingivally, or we can actually use paper points. We can insert them within the pocket, leave them there for a few seconds, and then collect them, transfer them into the tube, and then to the laboratory for various types of analysis. We can actually perform pure bacterial cultures, where we will be able to see only the viable cultural bacteria. We can perform real-time PCR analysis, which we can actually identify also the non-culturable bacterial by this approach, and is quite frequently used and established in various laboratories. Or we can perform fluorescence in situ hybridization or immunofluorescence using 16S rRNA targeted probes or antibodies against different species that we want to observe, and these are tagged with fluorochromes, and then we observe them under fluorescence microscopy to identify them. Of course, there is also a whole new world of identification with high throughput sequencing, more modern approaches, but I won't discuss those on this particular talk. This is a paper point, one of those ones, under a scanning electron microscopy, which has been used to collect a sample. You can actually see the fibers of the paper and in between the crevices. And when we take a closer look in the magnification here, you can actually see a whole lot of different microorganisms trapped within these crevices here. We have cocci, we have long rods, we have fusiforms, or spirochetic forms here. Spirochets are characteristic of deep periodontal pockets associated with periodontal disease. So we are actually able to trap microorganisms from the biofilm and transfer them in the tube. We can then vortex it and process further this sample for further analysis in the laboratory. I will give one example of the detection of one of these periodontitis-associated species, Porphyromonas gingivalis, perhaps the most well-studied putative oral pathogen. This is associated with chronic periodontitis, and it's considered it is a gram-negative anaerobic rod, so it grows particularly on an anaerobic environment, but it's also classified as a so-called black pigmented bacteroides. And it can produce a number of virulence factors, the most well-characterized ones being its cysteine proteinases, which are known also as gingipines, and they can actually cleave several proteolytic substrates. So if we look at a biofilm sample and we actually culture the sample anaerobically to detect the presence of P. gingivalis, you can actually see that it forms this nice black pigmented colonies on blood agar. P. gingivalis uses hemine from the blood substrate. It breaks it down in order to acquire iron for its own metabolism, and as a result we have the formation, we have this coloration. We can also look at under the microscope, under direct light microscopy here, you can see a dispersed biofilm. You have different morphotypes, different morphological shapes of different species. Again, we have some long rods, some fusiforms, and some cocci or small rods here and there, and some spirochetes. Then, when we perform fluorescence in situ hybridization analysis and look under fluorescence microscope, you can actually identify specifically where P. gingivalis is located. And you can actually see either single cells or clumps or aggregates of P. gingivalis. And this is pretty good proportion actually of microorganisms within a given sample. So P. gingivalis is highly represented in a biofilm sample of periodontal or chronic periodontal disease. But it's not always possible to study biofilms in vivo or clinically due to many technical limitations and ethical restrictions. So for these reasons, several laboratories throughout the world have been able to develop in vitro biofilm models where one can study in more close detail aspects of the etiology and pathogenesis of the disease. And the model I'm going to talk to you about is a model developed at the University of Zurich, originally by Professor Guggenheim approximately 15 or so years ago. And I have worked on this model. I've been working over the past 7 or 8 years together with my colleague Thomas Turner from the University of Zurich. So, how does the model look like? On one hand, we have to prepare pre-culture of different species of interest that we are about to mix. And on the other hand, we also have to prepare a salivary hydroxyapatite disc coated with a salivary pellicle. Hydroxyapatite is basically the mineral that constitutes the enamel of our teeth, and it's approximately 96% mineralized. And that is spontaneously covered with saliva as soon as the tooth erupts in the oral cavity. And it forms a thin layer of glycoproteins known as the salivary pellicle. And the bacteria are utilizing this pellicle to attach on the tooth surface. So we mix in different combinations the bacteria, 6 to 10 different species depending on the model that we're using, and we inoculate this mixed culture on a pellicle coated hydroxyapatite disc, and we can culture it aerobically or anaerobically depending on the species that are contained there. Change the media over the course of a few days, and we can harvest the biofilm at 64 hours or so. But there can be variations for longer or shorter incubation. So after 64 hours, that's what we get in our laboratory. You can see actually a 24-well plate here. Half of the plate is filled with hydroxyapatite discs on which biofilm has actually grown. And what can we do at this stage? There are a lot of possibilities. How do we process the biofilm? We can pick it up with a forceps. So actually this disc here contains on its surface a mature biofilm of the species that we have introduced. And we can do a number of things. We can actually quantify what is in there, each of the individual species at what levels are represented in this biofilm. Or we can perform confocal laser scanning microscopy, and we can actually identify where exactly each of these species is located within the biofilm. We can deduce different neighboring relationships between species in this manner. I'll describe briefly the variants of the biofilm models that we have used, and we have on one hand so-called supragingival species model, which consists of 5 bacterial species and 1 yeast that can commonly be found in a supragingival plaque. We have the subgingival model, consists of 10 bacterial species that can be grown anaerobically. And among those are the 3 so-called red complex species that have been considered to be, since the 90s, perhaps more than any other species associated with periodontal disease. But we know nowadays that these are important players, but the picture is even more complex than that. This is a more caries-associated biofilm model because it contains Streptococcus mutans, and when we feed this biofilm with sugar, it can produce a thick extracellular polysaccharide, and Streptococcus mutans is a well-characterized potential cariogenic pathogen. This we consider a more periodontitis-associated biofilm. And we have a further modification developed recently, the so-called submucosal biofilm, consists of 12 species. 10 of them are the subgingival species, but we have also introduced Staphylococcus aureus and Staphylococcus epidermidis, and we have cultured that on titanium surfaces. And the reason is that we wanted to develop a perimplantitis-associated biofilm. So dental implants consist of titanium, and titanium surfaces are also favoring the formation of biofilm. Quite a frequent find, or certainly more frequent than dental-related biofilms. Titanium-associated biofilms are one can actually find also staphylococci. That's why we decided to introduce them as well to answer questions related to peri-implant etiology and pathogenesis. Here are some examples of the use of these biofilm models in research. One can use these models to validate antimicrobial efficacy of different drugs. And then one study we have undertaken to compare the different antimicrobial efficacies of antibiotics commonly used in dental practice, actually in periodontal treatment as adjunct treatment modalities. And we used concentrations that are physiologically relevant. In other terms, concentration that one can find within the periodontal pocket. These concentrations are going to be to encounter the biofilm. So this we consider as relevant concentrations. We had metronidazole, amoxicillin, combination of those two, doxycycline, and more recently used azithromycin. You can actually see that these 4 last groups have caused it's a logarithmic scale here, have caused a significant reduction on the total levels of the bacteria present there. This is a subgingival biofilm, by the way, model. But I will go directly to the most striking differences, which are here the purple group and these 3 species, Fusobacterium nucleatum, Porphyromonas gingivalis, and Prevotella intermedia. Even if it didn't prove to be statistically significant, yet the numerical difference is quite pronounced. So these 3 species are in the list of putative periodontal pathogens, and they have dramatically their levels have dramatically been reduced by this antibiotic regimen, which is combination of amoxicillin and metronidazole. And we do know that this combination is perhaps yields the best clinical outcomes compared to other antibiotic regimens when they are used as adjuncts in periodontal diseases treatment. So this is basically a biological proof of the concept for the clinical observation of the high clinical outcomes of this antibiotic regime. Beyond the antimicrobial efficacy, we can also use biofilms to study the interactions between species within these communities. And I will present you part of a study where we actually wanted to find out the effect of gingipines, the virulence factor of P. gingivalis, on the biofilm growth and formation. We used P. gingivalis wild type, like we use in most biofilm cases, a lysine gingipine mutant strain, and then arginine gingipine mutant strain. These are 2 gingipines, the 2 primary gingipines of P. gingivalis. And first, so we generated 3 different biofilms and compared them. We looked at the number of each individual species within the biofilm, whether the wild type P. gingivalis was in, or the licensed gingival mutant, or the arginine gingival mutant. And we did not observe any striking numeric differences and non-significant differences for most of the species, with the exception of Tannerella forsythia, one of the 3 red complex species. When P. gingivalis was lacking the licence-specific gingipine, the numbers of Tannerella forsythia were significantly decreased. And this basically shows us an interdependency between forsythia and P. gingivalis. It looks like P. gingivalis supports the growth of Tannerella forsythia, mediated by the licence-specific gingipine. When this is removed, then Tannerella forsythia is not able to grow as well. So that's an example of, let's say, a symbiotic relationship between the two. We further looked in structural characteristics on other species, and we actually were able to find out that Treponema denticola in particular, when the wild type P. gingivalis was present, or when the licensed gingival mutant was present, they were Treponema was forming this kind of wheel-shaped bulky aggregates. But when the arginine gingival mutant was introduced, so when the arginine-specific gingival was missing, this conformation was lost. There was much more even distribution. So that also shows that the other gingival of P. gingivalis has a role in the functionality or in the functional arrangement of Treponema denticola. So far I've talked about biofilms on their own, but in order to understand the pathogenesis of the disease, one has to study biofilms in relation to the host. And we need, in these instances, to bring biofilms together with a cell type or a cell or a host tissue. Just a brief overview of periodontal pathogenesis at this stage. The bacteria contained in the biofilm, or the microorganisms of the biofilm, can actually stimulate an inflammatory response, manifesting as gingivitis. If the response is efficient, then it will actually result to control of the bacterial colonization and the restoration of health. But if the response is prolonged, then we will have pathogenic response. The inflammation will now become destructive, and the gingival connective tissue, as well as the alveolar bone, will be lost. So, how to study this type of interaction? We can bring together a number of cell types with the biofilm, like epithelial cells or fibroblasts, or indeed complex organotypic tissues generated in the laboratory. In this example, I give you some data from multi-layered gingival epithelium, which we cultured. And we have inserted, according to the standard model, a plastic ring in the culture. And on top of that, we placed the biofilm disc. We reversed it in such a manner that the biofilm surface is facing towards the epithelium. And that recapitulates, in a way, the interface between the tissue and the biofilm in real life. And when we perform a histology here, toluidine blue staining, you can see the control without the biofilm, multiple epithelial cell layers. With the biofilm, you can see that the biofilm has established over time, even on the epithelial cell surfaces. And one can study different things, like cytotoxic effects of the biofilm, or colonization or invasion, gene expression, and protein expression. I will show you an example of colonization. In this instance, you can actually see by scanning electron microscopy the epithelium, the outer surface of the epithelium, and we tested the effect of the red complex species on the colonization of the biofilm. So we generated a full biofilm of 10 species and a biofilm where the 3 red complex species had been removed, and we observed the colonization patterns. In the full control biofilm of 10 species, you can actually see bacteria colonizing the surface of the epithelium. When the 3 red complex species were taken away, you can see a dominance of bacteria on the surface. So there is a more pronounced colonization. When we take a closer look, we actually see that these are primarily coccoid cells, cocci, and we had basically streptococci in the biofilm. So we confirmed by confocal laser scanning microscopy and also by culture methods that these were indeed Streptococci and primarily Streptococcus oralis. Streptococcus oralis is a health-associated species and more of a commensal species. So you can actually see that when you take away the more pathogenic species, we have dominance of the more health-associated ones. So that also denotes a competitive interaction between different species in the biofilm here on the substrate of epithelial cells. We took this interaction model one step further, and we actually decided to find out what type of proteins are secreted by the epithelial cells when they are challenged, where they're infected by the biofilm. So we characterized the so-called epithelial secretome of the cells. And we considered the proteins that were more than twofold regulated in this respect. These are the differences of the upregulated and downregulated in response to the biofilm over 24 and 48 hours. You can actually see that over the upregulated proteins were 27 at 24 hours. The downregulated were more, where we had 64 proteins going down over 24 hours. At 48 hours, 18 proteins were upregulated and 125 were actually downregulated. When we compare these proteins between these two time points, 10 of those were commonly found in both time points. When we look at the downregulated ones, 62 of these proteins were common between the two time points. So the vast majority of the proteins that were downregulated here at 24 hours maintained at low level over 48 hours, and they were joined by another 63 proteins in this group of downregulated proteins. So it means that when these proteins are suppressed, they keep suppressed over a longer period of time. And that was quite interesting to see, because when we talk about responses during the early stages of infection, we usually consider what goes up but not what goes down. And that's an important indication that during the course of an infectious process, we also need to consider the proteins that are downregulated and what does this mean for the course of the development of the infection. When we cluster now these proteins into different, according to the different biological processes, based on the appropriate bioinformatics software, we actually found out that the upregulated proteins were associated with neutrophil aggregation, sequestering of zinc ion, chemokine production, leukocyte aggregation, and sequestering of metal ion. The downregulated proteins were associated more with cytoskeletal organization, epidermis development, skin development, actin filament-based processes, and actin cytoskeleton organization. And when we try to put all of these findings in a nutshell, one can say that the biofilm has affected the chemotaxis of the epithelium, the chemotactic properties of the epithelium, or the chemotaxis stimulating properties of the epithelium, has disrupted, the biofilm has disrupted the epithelial barrier, and that may also have implications in the impairment of wound healing. So to our view, this is like a molecular proof of principle of histopathological observations already from the 70s or 80s that actually demonstrate and describe such events during the early stages of periodontal inflammation or periodontal pathogenesis process. So at this stage, now I would like to have my concluding remarks. First, on oral biofilm research. One can say that the laboratory biofilm models can be effectively used to study interactions between species within the biofilm community, or their potential pathogenic interactions with the host, or they can be used in order to pre-validate different antimicrobial modalities before they are actually used in the clinic. High throughput methods nowadays are very important. They are valuable tools that need to be used in conjunction with biofilm models, and this will allow for the simultaneous collection of vast amount of data to be able to more globally characterize the events that take place during the early stages of inflammation and infection. And it's also very important to be able to clinically validate the findings and always question if there is relevance of these findings to the pathogenesis and the potential treatment of the disease. And if I may, like to make also some more general remarks about dental research at this stage. It's very important, as I mentioned, to clinically validate in vitro findings. We now need more longitudinal rather than cross-sectional studies. We've performed a lot of cross-sectional studies in dentistry. We actually need to follow results and data over longer periods of time. We need to study combinations of microbes and host factors, not like single bacterial or single host factors. We need to actually study signatures, as we tend to call them, of the disease, and that will be important for devising early diagnostic and prevention modalities. We need to consider novel agents in the treatment of oral diseases, primarily new antimicrobials, because there is with the increasing incidence of antibiotic resistance, we need to find other ways to use, other modalities to use as adjunctive treatments. We also need to consider agents that lead to resolution of inflammation, not only the eradication of the microbial causative factor, but also agents that support proactively the resolution of the pathogenic events. Perimplantitis is also a novel disease that we need to consider very seriously. Its occurrence is increasing by time, especially as we are increasingly using dental implants as treatment modalities in restorative and reconstructive dentistry. And last but not least, it's important also to consider the systemic impact of oral diseases and how does oral inflammation link to systemic inflammatory conditions like, for instance, diabetes mellitus or neurodegenerative diseases. And I'd like to conclude by acknowledging the different universities and colleagues in those universities that I have studied in or I have worked in, starting from the Aristotle University of Thessaloniki, where I obtained my dental degree, and moving shortly after that up to Umeå, where I did my PhD in oral microbiology. Moving down to London for a short postdoc in the beginning at King's College, and then at Queen Mary's University of London, where I attained my lectureship. And then moving to the University of Zurich, where I stayed 7 years. I attained there my docentship and my first professorship in oral microbiology and immunology, before moving a bit more than 1 year ago up here at Karolinska Institute. So, thank you very much for your attention. Questions?